In the previous video, we had an overview of the process of translation, that is the protein synthesis in prokaryotes. Now we will be dealing with the three basic steps of translation in a bit more detail. That is, we are going to understand the exact mechanism, what happens during initiation, then the elongation and the termination phase of translation. Basically, at the point of initiation, now since initiation is the point of a process in which the entire machinery it gets going. So for the purpose of translation, we can divide this process of initiation into activation of the various molecules involved. The activation or the charging of transfer RNA and the formation of the initiation complex. First the pre-initiation complex, and that will be followed by the formation of the initiation complex that comprises of the ribosome, the messenger RNA, and then it initiates the exact process of elongation, that is peptide bond formation between the amino acids. Now at the time of initiation of the translation process, first of all, the transfer RNAs that are present in the cell system, they have to get activated, they have to get charged, because we know that each transfer RNA has got a specific amino acid anticodon. So it is responsible for carrying a specific amino acid to the messenger RNA according to the codon present on the messenger RNA. So first of all, the first step is the activation of amino acid, you may say, or you may say the charging of the transfer RNA. Now this process in which the transfer RNA with its anticodon, it binds with the amino acid with the help of energy obtained from ATP, and this results in the formation of the amino acid transfer RNA or the activated or charged transfer RNA. So this entire process can be divided into two substages or two reactions. It comprises of two reactions, and both these reactions, they are catalyzed by an enzyme, that is amino acid transfer RNA synthetase. So what happens is, first of all, this amino acid, it gets acylated, that is the adenine group from the ATP molecule in the form of adenosine monophosphate that attaches with this uh, acid group of amino acid. So at this hydroxyl point, in first step, this adenine group is added. This results in the formation of adenylated amino acid, and the pyrophosphate molecule, that is the PPI, that is released. After this charged amino acid or this activated amino acid in the form of adenylated amino acid, it attaches to the 3' end of the transfer RNA. Now we know that transfer RNA is a polyribonucleotide sequence with the 5' and the 3' end, and because of the presence of complementary sequences within the structure, it takes the structure of the stem loop structure. This part of the transfer RNA has got the anticodon, which is complementary to the specific codon that is present on the messenger RNA, and its 3' end with the 3' hydroxyl group on the ribose sugar, it gets attached to this amino acid. So each transfer RNA, according to the anticodon present on it, it recruits a particular transfer RNA, and in this manner all the 20 essential amino acids, they are carried over by this adapter molecule, and it is carried over to the messenger RNA where polypeptide is formed or the protein synthesis takes place. So in the next step, this activated amino acid, that is adenylated amino acid, it forms the ester bond with the hydroxyl group present on the 3' of the ribose sugar in this transfer RNA molecule. This leads to the release of the adenosine monophosphate moiety, and this amino acid, it gets attached to the transfer RNA, and now this is a charged transfer RNA, or what we call as the amino acid transfer RNA. So one of the raw material is activated, that is the transfer RNA along with the amino acid. Now next, the raw material that is to be added upon, or that is to be organized, is the messenger RNA molecule. This messenger RNA molecule, in case of prokaryotes, it is present in the cytoplasm because no nuclear membrane is present. Organized nuclei, they are absent in prokaryotic cells. So the messenger RNA molecule that is transcribed on the genetic material, that is the DNA molecule, it has got certain specific regions. At the 5' end, we know there are certain UTRs, that is untranslatable elements or sequences. This is the leader sequence, and this 5' UTR has got a specific series, a consensus sequence of nucleotides, and that is known as the Shine-Delgarno sequence. This Shine-Delgarno sequence is basically responsible for positioning this messenger RNA in the protein synthesis machinery, that is the ribosome. So if we see this structure of messenger RNA molecule, in the leader sequence or the 5' UTR, there is the Shine-Delgarno sequence, and downstream this Shine-Delgarno sequence is your start codon, that is the AUG. Again, this is the consensus start codon in prokaryotes as well as eukaryotes. This is followed by the various nucleotides, the triplet codons for various amino acids, and it ends with the stop codon, which may be any of these three. This is followed by 3' UTRs, and in case of E. coli, it is this SECIS element. So these 3' UTRs, they will have their own role during the termination process. So the messenger RNA, it itself defines the initiation point, then the elongation will be carried over from the start codon to the stop codon, and once the stop codon is reached, termination of protein synthesis or translation will be there. So basically, what is this Shine-Delgarno sequence? In the messenger RNA, towards the 5' area region, there is a consensus sequence of GA, GG, AA, C, triple C. This is a consensus sequence, and of this consensus sequence, these nucleotides, the ribonucleotides in the messenger RNA, they determine basically the correct open reading frames for the mm, gene that follows, for the coding region that follows, because it defines the triplet, the frame of reading. And in this very open reading frame is present the start codon, followed by various triplet codons, and ending with the stop codon. Second thing is, this Shine-Delgado sequence, it provides the positioning of messenger RNA within the ribosome. We know that ribosome basically they are proteinaceous entities in which proteins they are associated with the ribosomal RNA molecules. So in the ribosome, there are two subunits, the small subunit and the large subunit. And we know that in prokaryotes, the smaller subunit is made up of 16S ribosomal RNA. Now, all this we have done in our introductory video on translation, which I recommend that you should view it before coming to this video on the mechanism of initiation. So, on the 16S ribosomal RNA, towards its 3' end, there is a complementary sequence, a sequence that is complementary for this Shine-Delgado element. So, this part of the ribosomal RNA, it forms bonds, or it forms the duplex with this Shine-Delgado sequence of messenger RNA. And this how the messenger RNA, it associates itself with the translation machinery, that is the ribosome. Now, once these ribosomal machinery, it associates itself with the messenger RNA at the Shine-Delgado sequence, that means now the elongation process will move downstream, that is from the 5' to the 3' region of the messenger RNA. Now we have the charged transfer RNA. We have got association of the messenger RNA with the ribosomal unit, 16S ribosomal RNA of present in the smaller subunit of ribosome. What remains is the association of this uh, ribosome messenger RNA entity with the charged transfer RNA. So, in the next step, various initiation factors, various proteins which help in this process of initiation, they get associated with the small subunit of ribosome. This small subunit of ribosome, it has got 3 sites corresponding to the 3 sites of the larger subunits. So since they are present on the smaller subunit, they are the E prime, P prime and the A prime site. So first of all, of the 3 initiation factors that are involved in initiation of translation in case of prokaryotic cells, first of all, it is this IF3, the initiation factor 3, which attaches itself near the E prime site of the smaller subunit. Now this IF3, it activates this uh, ribosome, which is comprised of 2 subunits, the small subunit and the larger subunit. Now, in the cytoplasm, the ribosome is in the form of our 70s ribosomal entity. Once this IF3, it attaches itself to the E prime site, near the E prime site of the smaller subunit of this ribosome, it disassociates the 50S subunit of the ribosome. And this is assisted over by another initiation factor, that is IF1. This IF1, it attaches itself to the amino acid site, corresponding amino acid site of the smaller subunit. That is, it associates itself with the A prime site of this uh, smaller subunit. So E prime, it does not permit the complementary reaction at this E prime site. This IF1, it blocks the A prime site, and what remains is the P prime site. Now we know that this messenger RNA, it is present associated with this smaller subunit. In case of prokaryote, first this smaller subunit, it gets associated with the messenger RNA, and then after your charged transfer RNA, it comes and occupies this peptidal site. So this charged transfer RNA, charged with formyl methionine. This formyl methionine containing transfer RNA, it is associated with another initiation factor, that is IF2. Now this IF2 is a GTPase protein. That means it is associated with the GTP molecule, the guanine triphosphate molecule, which again is the energy currency. So along with its IF2, this IF2, it basically guides this transfer RNA that is charged with this formyl methionine, and it brings it so that it may associate with the P site. The E site is blocked, the A site is blocked, and for the purpose of initiation, only P site is available. So, and this P site, it has got the AUG sequence of the messenger RNA. One more thing we should understand at this point is that within the coding sequence, AUG may be present at many points, but the one that follows the Shine-Delgarno sequence, that is the start codon. So only that is the point of messenger RNA that comes to lie in this P prime region of the smaller subunit with its codon AUG in this particular region of the smaller subunit. And the charge transfer RNA with the formyl methionine, it is the only transfer RNA which comes and it associates itself with the P site, because when we'll see the elongation part, we'll see that all other charged transfer RNAs, they'll first come and associate with the amino acid site. But this formyl methionine, it associates with the P site. So this pre-initiation complex is formed. In this pre-initiation complex is the initiation factor 1 and 3 blocking the E prime and the A prime site, and there is this initiation factor 2 with its GTP, and at the P site your charged transfer RNA is there. The messenger RNA, which enters this ribosomal small ribosomal subunit, sorry, ribosomal smaller subunit, it is present in the cleft of the ribosome, so that it is exposed to these sites where further these charge transfer RNA, they will come and enter. So this is your pre-initiation complex. Formation of this pre-initiation complex is then followed by the addition of the larger subunit of the ribosome. Now, this is activated, or this is made possible by the hydrolysis of GTP molecule. This GTP, it hydrolyzes itself to form GDP, and once it hydrolyzes itself to form GDP, the various transcription factors, that is the transcription fact sorry, not the transcription factors, the initiation factors, the initiation factor 1, the initiation factor 3, and also the initiation factor 2. All these initiation factors, they are released, and we know that basically as soon as this initiation factor 3, it is released, the larger subunit or the 50th subunit, it associates itself with its corresponding smaller subunit. Now these three sites of the ribosomal entity, that is the E site, P site and the A site, the amino acid site, the peptide site and the exit site, they come to lie corresponding to the various E prime, P prime and A prime sites, and thus our initiation complex is in place with the first amino acid, that is the formilated methionine, attached to the start codon, and the A site is available for the next charge transfer RNA corresponding to the triplet code present at this site. Now, during all these stages, whenever any initiation factor, it associates itself with the ribosome, conformational changes, they take place. So this riboproteinaceous entity, that is the ribosome, it is a dynamic structure, and once it gets associated with the messenger RNA, gradually it will move along from the 5 prime to the 3 prime region, and once it will start moving, there will be movement of this, or different triplet codes, they will come to occupy these particular sites. So that we will be dealing with during the next step of our translation mechanism, that is the elongation part.